Vanakkam. The beauty of the nerve lies less in the anatomy and more in the functioning. And the anatomy helps it in every way. When we make an attempt to understand the basic function of the nerve, we will realize that ultimately it is the development of the action potential of the nerve that is the sheet anchor. How it develops, how it is propagated, and how it achieves the end result, whether the sensory function or the motor function, and most importantly, why we need to understand it. All this explained in a simple method in this video. Our agenda today on learning about the physiology of nerve conduction will encompass the understanding of the resting membrane potential, the method by which the action potential is initiated, and the factors involved in the initiation of this action potential of the nerve, how this action potential is propagated along the length of the axon, the different factors which determine the speed of this propagation, the neuromuscular junction, and finally the physiology of the development of the compound muscle action potential and the sensory nerve action potential, along with the clinical applications of all these topics. First, we shall see the resting membrane potential. Let us consider a few facts before we go into the development of the resting membrane potential. Neurons, like other cells, are more negatively charged inside than outside. This results in a membrane potential of about minus 70 millivolts. This is called the resting potential of the neuron. In this state, the neuron is said to be polarized because it has got a negative charge on one side and a positive charge outside. At the same time, this resting potential will not last because the neurons are excitable cells, and when the membranes of the neurons become depolarized, the neurons get into the excited state. The neuron has a cell membrane and a cytoplasm which is known as the axoplasm. The external area is filled with extracellular fluid. The exterior surface contains mainly sodium, calcium and chloride ions, but it is mainly positively charged, whereas inside the cell are mainly potassium and other negative anions, so the inside of the cell membrane is negatively charged. Now, if we are able to measure the electrical difference between the inside and the outside of the cell, we find that there is a voltage of minus 70 millivolts. This is the resting membrane potential of the neuron, also known as RMP. This is the resting state. When it is stimulated, the action potential forms. The action potential is a sudden, fast, transitory and propagating change of the resting membrane potential. It has got three stages: the stage of depolarization, the stage of repolarization and the stage of hyperpolarization, and there are refractory phases in the action potential. So, when there is an electrical stimulus, it acts on the resting membrane potential and then it reaches the threshold potential, after which the action potential is initiated. We have seen that the electrical charge of the cell is determined by the ions within it and outside it. The potassium inside gives it a negative charge. The sodium outside gives it a positive charge. So, if we can move the ions, we can get an electrical stimulus to form. Let us now consider the neuron in the resting state with the resting membrane potential of minus 70 millivolts. In the cell membrane, we have got different channels for different ions. The sodium channels, the potassium channels, the chloride channels, and the separate sodium-potassium pump. On a stimulus reaching this neuron, it leads to the opening of the sodium channels. When the sodium channels open, the sodium ions which are concentrated outside the cell rush into the cell through these specific sodium channels. When there is a buildup of sodium ions inside the cell, you will note that the electrical charge on the inside has now become positive and the electrical charge on the outside has become negative. So the neuron is now depolarized. But if the electrical gradient is measured, it would be about minus 60 millivolts, and this cannot initiate an action potential because the threshold for action potential initiation is minus 55 millivolts. So this stimulus has been a sub-threshold stimulus. Since there has been an influx of positive ions, the resting membrane potential of minus 70 millivolts is now reduced and has become minus 60 millivolts. This influx of positive ions has caused the excitation of the nerve, so it is called excitatory postsynaptic potential or EPSP. On the other hand, if yet another stimulus causes the opening up of the chloride channels alone, the chloride ions would move in, bringing more negative charge into the cell. Since it has become more negatively charged, the electrical gradient would be now around minus 80 millivolts. The neuron is now said to be hyperpolarized. Since there has been an influx of negative ions, the resting membrane potential of minus 70 millivolts is increased, has become minus 80 millivolts. This influx of negative ions has caused the inhibition of the nerve, so it is called the inhibitory postsynaptic potential or IPSP. These changes in the electrical potential and the state of the nerve have been due to the movement of ions through the gates. So, what controls these gates? There are ligand gated channels, voltage gated channels and stress activated channels. Ligand refers to the chemicals or neurotransmitters. Some chemical will cause the gates to open, ions to influx and action potential to be initiated. The typical example being the neuromuscular junction where acetylcholine is the neurotransmitter. Some gated channels are controlled by the electrical stimulus which causes the gates to open, ions to influx. These are called the voltage gated channels, and the best examples are the voltage gated sodium channels and the voltage gated potassium channels seen on the cell membrane, and is responsible for the propagation of the action potential in the axon. The third type of control are the stress activated channels. Stress like pressure, temperature will cause the gates to open, ions to influx and action potential to be initiated, as is seen in the sensory nerve endings. The action potential can be generated only when the stimulus changes the membrane potential to the values of threshold potential, which is around minus 55 millivolts. And here the all or none law comes into play. Any subthreshold stimulus will cause nothing, while threshold and supra-threshold stimuli produce a full response of the excitable cell. There are three methods by which the electrical stimulus that comes in can reach the threshold and cause the action potential to be initiated. The first is a single supra-threshold stimulus, second is summation of multiple stimuli, and third is balance of EPSPs and IPSPs. We saw the example of a sub-threshold stimulus which reached an electrical gradient of minus 60 millivolts, so an action potential could not be initiated. However, if the single stimulus could be a supra-threshold stimulus, it could cause the threshold to be achieved. This will initiate the action potential. This single stimulus cannot reach the threshold, but multiple small stimuli at different points along the cell body can cause the threshold to be achieved. This is called spatial summation, as it comes from different points. A single stimulus at one point may not be able to cause an action potential, but if there are multiple recurring stimuli at the same point within a short time of each other, it can get summated and cause the action potential. This is called temporal summation. We have already seen the EPSPs and the IPSPs. The EPSPs are positive stimulations while the IPSPs are negative stimulations. When both the EPSPs and the IPSPs are acting in large numbers, The sum total will decide the strength of the stimulation, whether it is sub-threshold or supra-threshold. When the depolarization reaches minus 55 millivolts, the threshold is reached and the action potential is initiated. Once the action potential has been initiated, two things should happen now. The segment where the action potential was initiated should get back to the resting state, and secondly, the initiated action potential should propagate forward. Regarding the first event of getting back to the resting membrane potential, two events are involved. The voltage gated potassium channels cause the potassium to leak out, leading to a reduction in the positive charge inside the cell, and active sodium potassium pumps push the sodium out, restoring the negative charge inside the cell. After the depolarization has occurred, the voltage gated potassium channels open, causing a slow influx of potassium ions outside. Now you will note that the electrical gradient has come to minus 60 millivolts. Now there is still a problem. The potassium has gone outside, the sodium is still inside, electrical potential is not completely restored, the sodium and potassium ions balance must be restored, and sodium must go out and potassium must be brought in against their gradient. This is possible by the sodium potassium ATPase pump. So now the active sodium potassium pumps open and push the sodium out. For every 3 ions of sodium which are pumped out, 2 ions of potassium are brought inside the cell. This pump is not a passive channel but an active channel which uses ATP for this activity. Now the resting membrane potential of minus 70 millivolts is restored and the ion balance is also restored. Now we shall review the sequence of this action potential. The neuron first in the resting phase with an electrical gradient of minus 70 millivolts. A small sub-threshold stimulus is not strong enough to reach the threshold of minus 55 millivolts. Hence the neuron continues in the resting phase. The next strong stimulus, which is a maximal stimulus, reaches the threshold of minus 55 millivolts and hence triggers the depolarization and the action potential. This is the depolarizing phase in which it can reach up to plus 30 millivolts. Then it starts repolarizing again in the now called repolarizing phase. This repolarization does not immediately stop at minus 70 millivolts but goes beyond that, hence called the hyperpolarizing phase. The electrical potential then comes back to minus 70 millivolts, which is the resting membrane potential, and enters the resting phase again. When the nerve is going through the action potential phases, it cannot be stimulated by any other stimulus coming in. This is because it is refractory to another action potential since the sodium channels are inactive and need to be in a closed state. This stage ends when enough sodium channels recover from their inactive state. The absolute refractory period is when even a very strong stimulus cannot initiate an action potential, and the relative refractory period is when a supra-threshold stimulus can start another action potential. This period of depolarization and repolarization is the absolute refractory period, and the period of hyperpolarization is the relative refractory period. The second thing that the action potential has to do is propagate forward. We shall now see how it propagates forward along the axon. The action potential summation and initiation usually occurs at the axon hillock. This propagates along the axon. It is called the continuous conduction. We shall now see how this conduction actually occurs. Let us assume that this is the neuron with the negative charge inside and the positive charge outside. When the action potential is formed, we know that the positive charge comes inside and the negative charge goes outside. This itself is an electrical stimulus and causes the voltage gated sodium channels on the neighboring segment of the neuron to depolarize, and hence the action potential shifts to that segment. Now the action potential cannot go in a reverse direction because that segment is in the refractory period. Hence the impulse can only move forward. As the impulse moves forward, the first segment now comes into the resting phase. This impulse propagates till the terminal end of the axon. However, this type of continuous conduction is very slow, especially when we consider that some axons are very long, especially those that come from the spinal cord to the feet. The method of nerve impulse propagation in the myelinated nerves is different. The same nerve impulse or action potential that has been initiated in the axon hillock travels first to the unmyelinated portion of the myelinated neuron. This nerve impulse does not travel under the myelinated portion of the nerve. It jumps over the myelinated portion to the next node of Ranvier, which has a high density of voltage gated ion channels, and that segment is depolarized next. This type of conduction is called saltatory conduction and is much faster than the usual continuous conduction that we have seen. As we can see, this fast method of conduction is possible only in the myelinated neurons. The features of this saltatory conduction are that no depolarization occurs under the myelin and depolarization occurs only at the nodes. All the sodium channels are concentrated at the nodes. In the myelinated nerves, the triggering jumps from one node to the next, and this, as we have seen, is much quicker than depolarizing all the membrane along the whole axon. There are some factors which can determine the speed of this propagation. The first factor we have already seen, that is the amount of myelination. The second factor is the axon diameter, and the third factor is the temperature. As we have seen, the myelinated fibers have faster conduction, whereas the unmyelinated fibers have slow conduction. As far as the axon diameter is concerned, the larger the diameter, the faster will be the conduction, and smaller the diameter, slower the conduction. Ambient temperature is ideal for a good nerve conduction velocity. The conduction velocity increases by 5% per degree Celsius as the temperature of the nerve increases from 29 to 38 degrees Celsius. Extremes of temperature slow down the conduction speed. All these features have been incorporated in the classification of nerve fibers, the Erlanger and the Gasser classification, which goes by the letters A, B, C, and the Lloyd and Hunt classification, which goes by the numerals 1, 2, 3 and 4. Type A fibers are subclassified as A alpha fibers, which are mainly somatic motor and somatic sensory, A beta fibers, which are mainly for the vibration sense, proprioception and light touch, A gamma fibers, which are mainly for the sensations of touch and pressure, and the A delta fibers, which are important for pain, crude touch, pressure and temperature. Type B fibers are mainly the preganglionic autonomic fibers, which have a conduction velocity of 3 to 14 meters per second, and the C fibers, which are unmyelinated, can be sensory for pressure, slow pain, crude touch and temperature, or autonomic, which is mainly for the postganglionic sympathetic fibers. As you can see, these fibers being unmyelinated are very slow conductors. These fibers have different susceptibility to different agents. For instance, when there is a nerve blocked by hypoxia, the most susceptible are the B fibers, followed by the A and lastly by the C fibers. As far as susceptibility to pressure is concerned, the most susceptible are the A fibers and least susceptible are the C fibers. When blocking with local anesthetics, the most susceptible are the C fibers, which are unmyelinated, and the least susceptible are the A fibers. There is a decreased conduction velocity in demyelination or compression or traction injuries of the nerve, and a total conduction block in complete transection of the nerve. When there is demyelination or compression, it causes disturbances in the microcirculation, affecting the permeability of the axon basement membrane. This results in a conduction block, and the flow of axoplasm is also interrupted. Whereas in a complete transection of the nerve, there will be a total conduction block and no impulse transmission is possible. As far as the motor nerves are concerned, the action potential traveling along the axon reaches the neuromuscular junction. Generally, there are two types of synapse, the electrical synapse and the chemical synapse. In the electrical synapse, the action potential moves directly through what is known as a gap junction. This is usually found in the heart and smooth muscle. The second type of synapse is the chemical synapse, where it is separated by a synaptic cleft. This synapse could be axodendritic or axosomatic or axonic. Here, the neuron sending the signal is present in the presynaptic region. The presynaptic region contains synaptic vesicles, which are sacs in the synaptic end bulb containing the neurotransmitter acetylcholine. The cell receiving the signal is the postsynaptic cell. This is the motor end plate as far as the muscle is concerned. It is the region of the muscle cell membrane opposite the synaptic end bulbs with acetylcholine receptors. When the action potential travels down the motor neuron and enters the motor end plate, a sequence of events occurs. When the nerve action potential reaches the presynaptic area, depolarization occurs due to the sodium influx, as we have already seen. This causes the opening up of what are known as the calcium channels, and as a result, calcium enters the presynaptic area of the nerve. This triggers the release of acetylcholine from the vesicles which are present in the presynaptic area. The acetylcholine is released into the synaptic cleft. This acetylcholine, which has been released into the synaptic cleft, binds to the post-junctional receptors in the motor end plates. These motor end plates are found in the muscle, and this leads to the opening up of the sodium channels. And as we know, opening up of sodium channels leads to depolarization and the generation of an action potential. In the nerve, this action potential, after being generated, would travel along the axon, but this being the muscle fiber, the action potential is transmitted along the sarcolemma to open up tubular calcium channels again and results in muscle contraction. When there is a muscle contraction, just like in the nerve, there is an electrical activity that is recorded. This can be recorded in the nerve conduction study. This is called the compound motor action potential, the CMAP, which represents the summation of all the recorded individual muscle fiber action potentials. In this recording, the latency, the amplitude, the duration, the CMAP area, and the nerve conduction velocity are measured. This diagram shows the typical CMAP. The latency is the time from the stimulus to the initial CMAP deflection from the baseline. Three processes occur during this latency period: the nerve conduction time from the stimulus site to the neuromuscular junction, the time delay across the neuromuscular junction, and the depolarizing time across the muscle. All these are measured in the latency. Next to be measured is the amplitude. It is measured from the baseline to the electrical negative peak, which is usually seen above the baseline. This may be lowered in conditions like axonal neuropathy, demyelination with conduction block, and presynaptic neuromuscular junction disorders. After measuring the amplitude, we measure the duration. This is measured from the initial deflection from the baseline to the first baseline crossing. The duration is increased in demyelinating diseases. The area of the CMAP, that is, the area measured between the baseline and the negative peak, is also very important. The differences in CMAP area between the distal and proximal stimulation sites signify conduction block from a demyelinating lesion. And finally, the nerve conduction velocity can be calculated. This measures the speed of the fastest conducting motor axons. It is calculated in meters per second. It is calculated by subtracting the distal latency from the proximal latency and divided by the duration. A conduction block in a nerve occurs either due to an acute injury due to trauma to a nerve, which is a blunt trauma causing neuropraxia, or a chronic injury like a entrapment neuropathy.
The definite features of a conduction block on nerve conduction studies are more than 50% drop in the CMAP amplitude with less than 15% prolongation of the CMAP duration, or more than 50% drop in the CMAP amplitude and area, or more than 20% drop in the CMAP amplitude and area over a short nerve segment of about 10 centimeters. This shows an example of a conduction block in the median nerve after being stimulated from the wrist, the elbow and the axilla. This shows the reduction in the CMAP amplitude and consequently a reduction in the area of the CMAP. Just like recording the nerve potential in the motor nerve, a sensory nerve action potential can also be recorded. This is a compound potential that represents the summation of all the recorded individual sensory fiber action potentials. Here we would be measuring the onset latency, the peak latency, the duration and amplitude, which are important. This shows a typical sensory nerve action potential recording. The onset latency is the time from the stimulus to the first deflection from the baseline. It represents the nerve conduction time from the stimulus to the recording electrodes for the largest cutaneous fibers. The peak latency, on the other hand, is measured at the midpoint of the first negative peak, which occurs above the baseline. The duration is measured from the onset of the potential to the first baseline crossing. This duration is much shorter than the CMAP duration. And finally, the amplitude, which is measured from the baseline to the negative peak. Low SNAP amplitude indicates definite disorder of the peripheral nerve. Again, the sensory nerve conduction velocity is measured by dividing the distance travelled by the onset latency. The sensory nerve action potential is actually more significant than the CMAP because these have a smaller amplitude. They are more sensitive for detecting mild or early disorders, and the sensory fibers have a lower threshold to stimulation than the motor fibers. So we need to remember that the sensory nerve action potential remains normal in lesions proximal to the dorsal root ganglia, even if there is a loss of sensation in that area which is being tested.
I hope you liked the video. Please click on the shown links to see more about the basics of nerve repair and reconstruction, including a detailed anatomy. And do not forget to subscribe to keep connected with the latest in learning hand surgery.